Good evening, guys. As promised, here's a video on some stuff we talked about in class yesterday and today. So we're going to combine the spontaneous generation experiments with kind of how scientists work. So let's get started. I'm going to go kind of fast so the length of the video is not long. Of course, you always have the option of stopping and starting. So going back and looking at spontaneous generation again, or abiogenesis as we called it yesterday in class. Four classic experiments starting in the 1600s. There was a common belief that life could come out of lifelessness. Out of non-living matter could produce things that were alive. And we know today that's kind of ridiculous, but back then people believed that. Well, except for one guy, Francisco Redi, he didn't believe it. So in the 1600s, he set out to prove that it couldn't happen. So he used jars, rotting meat, covers for the jars, and flies or maggots. So the maggots was his living thing, and the rotting meat was his non-living thing. So what he did was he had one group of jars with meat in it that were not covered, another group that jars of meat that were covered. And you can see in the picture he had two different kinds of covers, but it doesn't matter because both covers prevented flies from reaching the meat. They couldn't lay eggs, maggots couldn't form. So therefore this proved spontaneous generation, or so we thought. 70, 80 years later in the next century, an Englishman named John Needham thought maybe spontaneous generation could still occur and went out to try to prove it. Instead of maggots and meat, he used chicken broth and bacteria. Bacteria was the living thing, chicken broth was the non-living thing. He had a flask, it's like a laboratory container, and he basically did this. He boiled the broth to kill the bacteria. He left one flask open, sealed the other flask. The open flask, of course, grew bacteria, but so did the sealed flask. So he thought he had successfully proved that spontaneous generation could occur. 7 years later, an Italian guy, Lazzaro Spallanzani, had taken a look at Needham's experiment and realized he thought he made a serious error. So he repeated the experiment. Go back and look at the experiment if you need to. The big change he made was a little change, but it had big effects. Was he sealed the jars first, then boiled them to kill the bacteria. So I guess he hypothesized that after Needham had boiled the bacteria and then sealed the flask, then bacteria got in the time it took him to seal the flask. Okay, so in this case, he sealed them, boiled them, and then no bacteria formed inside the sealed jars, or sealed flask, I'm sorry. So this helped disprove spontaneous generation. So at this point, you have Redi and Spallanzani both disproved it. Needham thought he had proved it, but he really didn't. So people were still arguing the next century. Scientists were still not convinced of this. They argued that Spallanzani, by sealing the flask, prevented air from getting to the broth. And at the time, they had this belief that the air was the vital force. Okay? So the vital force, that means the life force. So they said, no wonder no bacteria grew, because you didn't let air in. So Louis Pasteur, the person we get to give credit for pasteurization process of milk and juice and other things, he said, okay, I'm going to design a new flask. I'm going to design a flask that will allow air in, but I think we'll keep the microorganisms, the bacteria, out. He did just that. So in this, you see in the picture there, he boiled the broth or the gravy in two flasks. He broke the neck off of one of the flasks, thus created an opening for bacteria, and of course bacteria grew. But the other flask, I read somewhere, reportedly stayed open to the air, that the air could actually travel through the neck of the flask, but not the bacteria. Remember, bacteria can't walk, fly, or swim. They have to be carried by air currents. And I guess this S-shaped neck trapped the bacteria and didn't allow it to make it to the broth. So no bacteria grew, but I read that he kept this open for a whole year and no bacteria grew. So this, for once and for all, put the idea of spontaneous generation to rest. So switching gears, and we'll try to use the experiment that we used in class. Now, however, I know we didn't have second period today, so second period guys didn't get this. So why do scientists do experiments? Why do scientists ask questions? And the obvious answer is to get answers. Okay? So when you think about asking questions, you're trying to find out the who, the what, the where, even the why, the how. Okay? And before you ask a question that you may test scientifically, you really need to make sure the question is refined. You want to make sure that it doesn't have too many unnecessary parts or words. You also want to make sure it's also asking a question about one specific thing only, and not a bunch of things. Okay? So we use the example in class. Do coffee grounds help plants grow? Well, after you've clearly defined your problem, it's time to form your hypothesis. And a hypothesis is a predicted answer to the problem or question, or a proposed answer or problem. A proposed answer to your question. Either way, you're trying to predict what's going to happen before you find out what's going to happen. Okay? So, remember, it's not a guess, not even an educated guess. So the example we used in class today was coffee grounds will help plants grow. Now, I could have chosen will not help plants grow, but I'm going with will. So all I did was basically take my problem, which was a question, and turn it into a statement for my hypothesis. So now it's time to test that hypothesis. We call a test of a hypothesis an experiment. In order to do an experiment, you have to come up with a set of materials and a list of procedures to use those materials. It's kind of like a recipe. What kind of ingredients did you use to bake the cake? And then what did you do with those ingredients when you put them together? Did you mix this and beat that and bake that and cook that? That's your procedures. So the steps you took to get there. An experiment must only test one thing at a time. If you want to test two things, you need to break it down and pick one, and then do a second experiment to test the other. But an experiment can only test one thing at a time. Otherwise, you wouldn't know which thing actually caused the event to happen. There's no way to tell the difference. So in an experiment, so this is still the experiment continued. In an experiment, you have things called variables. There's two very important variables and then another kind of variable. The independent variable is probably the most important thing because it's what you're testing. It's what you're trying to see if it makes a difference or not. So in this experiment, our independent variable is the coffee grounds. Okay? The dependent variable is what you're going to measure to see if the coffee grounds, in this example, make a difference or not. So the dependent variable is measured to see if the independent variable made a difference. Now finally, control variables, and there can be many of these. Basically, they're not like the other two variables. These are controls that you want to put in to make sure that all other parts of the experiment that you're not testing, in other words, not the independent variable, the control variables are those things you want to keep the same, constant. For example, in the coffee ground experiment, you wouldn't want to use different brands of coffee. You wouldn't want to use different amounts of coffee. You certainly wouldn't want to use different plants. Okay, you want to keep everything the same, except some plants get coffee grounds, some plants don't get coffee grounds. It's that simple. Also in your experimental section, you're going to end up collecting data. The experiment's going to produce the data. And you're going to collect it, write it down, you might organize it, you might make a nice graph or a chart, and then you're going to take a good hard look at it. Two kinds of data. Quantitative, which means numerical or numbers. Examples I gave you, plants grew 14 inches. Another example, the mass dropped 40 grams. Or qualitative data. These are descriptive words. These are adjectives. It's going to describe something. The food tasted bitter. The solution turned blue. Are examples of qualitative data. After you've got your data, you're going to draw a conclusion. You're going to figure out, was I right? Does the data show I'm right? Okay? So after looking at your data, ask yourself the all-important question, does the data support my hypothesis? So in our case, if the plants that received the coffee grew more than the plants that didn't, then our hypothesis was right, and the data supports that. If not, if the data didn't support it, then you got to make a decision. Do you just quit and give up? Or do you go back to step 2, write a new hypothesis, step 3, conduct a new experiment, step 4, get new data, and do it all over again. Okay? That's pretty much it. Be sure you take good notes. And that's it, and I'll see you tomorrow in class.